This method can help answer key questions in the geomechanics field about compaction processes in rocks.
The main advantage of this technique is that it allows quantitative measurement of the local elastic field within rock and mineral aggregates.
Though this method can provide insight into stress distribution within samples during cold compression, it can also be applied to other techniques, such as high-temperature compression.
Generally, individuals new to this method struggle because of challenges in preparing the sample and the cell assembly.
Sample and cell assembly preparations are difficult to learn without visual demonstration because of how small and delicate the individual samples and parts of the cell assembly are.
To begin preparing a mineral aggregate sample, grind about 15 grams of a rock specimen or a pre-existing powder to approximately 4-micron diameter grains using a mortar and pestle or a rotary tool with a grinding head. Pour the mineral grains into a 20 centimeter decantation column containing ethanol. Allow the grains to settle for an appropriate duration for their density. Then partition the suspension by height into 3 beakers. Allow the contents of the beakers to dry in air overnight. Measure the average grain diameter of each batch and select the batch in which the average grain diameter is closest to 4 micrometers. Next, grind the ends of an alumina rod to be flat and parallel within 0.5 degrees. Clean the rod and an alumina ring by sonication in ethanol for 10 seconds.